Hello students, today's topic of discussion is digestion and absorption of nutrients. The gastrointestinal system is the portal of entry of the nutrients, vitamins, minerals, and fluids. Carbohydrates, proteins, and fats are broken down into absorbable units, principally in the small intestine. The products of digestion and the vitamins, minerals, and water cross the mucosa and enter the lymph or the blood. But today we discuss digestion and absorption of carbohydrate, protein, and fat in this lecture. Digestion of carbohydrates. The principal dietary carbohydrates include polysaccharides, disaccharides, and monosaccharides. The starches and their derivatives are the only polysaccharides digested in human gastrointestinal system. In glycogen, the glucose molecules are mainly in the straight chains, that is, 1,4-alpha linkages, but few branching is also present, that is 1,6-alpha linkages. Amylopectin constitutes 80 to 90% of dietary polysaccharides, but is less branched. But amylose is a straight chain with 1,4-alpha linkages. Glycogen is of animal origin, whether amylopectin and amylose are of plant origin. The disaccharides include lactose or milk sugar, sucrose or table sugar, whether the monosaccharides include fructose and glucose. In mouth, the enzyme alpha amylase present in the saliva acts on the starches. The optimum pH of this enzyme is 6.7. Its activity is inhibited by acidic gastric juice in stomach. In small intestine, both the salivary and the pancreatic amylases act on the ingested polysaccharides. Both the salivary and the pancreatic alpha amylase hydrolyze 1,4-alpha linkages, sparing 1,6-alpha linkages, the terminal 1,4-alpha linkages, and 1,4-alpha linkages next to the branching points. The end products of digestion by alpha amylase are maltose, the disaccharide, maltotriose, the trisaccharide, glucose oligomers, and alpha-limidextrin, the polymer. This picture shows the structure of alpha-dextrin, maltotriose, maltose, and the disaccharides lactose and sucrose. The oligosaccharides are present in the outer portion of the brush borders, the membrane of the microvilli in small intestine. The oligosaccharides include alpha-dextrinase, isomaltase, maltase, and sucrase. Some of these enzymes have more than one substrate. Alpha-dextrinase or isomaltase mainly hydrolyzes the 1,6-alpha linkages present in alpha-dextrins. Alpha-dextrinase or isomaltase, in association with maltase and sucrase, breaks down maltotriose and maltose. There are also disaccharides present in the membrane of the microvilli in small intestine, which include trehalase and lactase. Lactase hydrolyzes lactose into glucose and galactose, whereas sucrase hydrolyzes sucrose into glucose and fructose. Deficiency of one or more than one of these oligosaccharides may cause diarrhea, bloating, and flatulence. Diarrhea is due to presence of increased number of osmotically active oligosaccharides in the intestinal lumen, causing the volume of the intestinal contents to increase further and thereby causing diarrhea. Absorption of carbohydrates. Hexoses and pentoses are rapidly absorbed across the mucosal cells in small intestine. The transport of hexose is dependent on sodium ion concentration or the amount of sodium ion present in the intestinal lumen. Both sodium and glucose share the same co-transporter or same port named sodium-dependent glucose transporter. There are two types of this import. One is SGLT1 and another is SGLT2. Sodium ion concentration inside the enterocytes is low. Therefore, sodium moves along its concentration gradient into the enterocytes. Glucose moves along with sodium and is released inside the enterocytes. Sodium is actively transported into the lateral intercellular space, utilizing the energy. Glucose is transported out of the enterocyte by a transport system named GLUT2 present on the basolateral membrane of the enterocytes into the interstitium, then into the blood of capillaries draining into the portal vein. The transport of glucose across the basolateral membrane of the enterocytes is an example of secondary active transport. The energy for the transport of glucose across the basolateral membrane of the enterocytes is derived indirectly from active transport of sodium into the lateral intercellular space. The glucose mechanism also transports galactose. When there is congenital defect in sodium glucose cotransporter, results in glucose or galactose malabsorption, causes severe diarrhea that is often fatal. The end products of digestion of sucrose are glucose and fructose. The end products of digestion of lactose are glucose and galactose. Both glucose and galactose utilize the same co-transporter, sodium-dependent glucose transporter, for their transport into the enterocytes. Fructose is transported by a different mechanism which is independent of sodium or transport of glucose or galactose. Rather, fructose is transported by facilitated diffusion by GLUT5 present in the apical membrane of the microvilli of small intestine into the enterocytes, and from there fructose is transported out of the enterocytes by GLUT2 present on the basolateral membrane of the intestinal mucosal cells. Protein digestion. Protein digestion begins in the stomach, where pepsins cleave some of the peptide linkages. Pepsins are secreted in form of inactive precursors called pepsinogens and are activated in the gastrointestinal system by gastric hydrochloric acid. Pepsinogens are of two types: pepsinogen 1, which is found in acid-secreting regions, and pepsinogen 2, which is found in pyloric regions of stomach. Pepsins hydrolyze the bonds between the aromatic amino acids phenylalanine or tyrosine with a second amino acid. The products of peptic digestion are polypeptides of diverse sizes. Pepsins have an optimal pH of 1.6 to 3.2. Their action is terminated when the gastric contents are mixed with alkaline pancreatic juice in duodenum and jejunum. The pH of intestinal contents in duodenal cap is 2 to 4, and pH in the rest of the duodenum is about 6.5. Trypsin, chymotrypsin, and elastase act at interior peptide bonds of polypeptide molecules. They are called endopeptidases. This picture depicts formation of active endopeptidases from their precursors in a chain reaction. Carboxypeptidases of pancreas are exopeptidases which hydrolyze amino acids at carboxyl and amino terminal of polypeptide molecules. Trypsin, chymotrypsin, and elastase act at interior peptide bonds of polypeptide molecules, and they are called endopeptidases. Whether carboxypeptidases of pancreas or exopeptidases which hydrolyze amino acids at carboxyl as well as amino ends of the polypeptide molecules. Some free amino acids are liberated in the lumen of the small intestine, whether other amino acids are liberated at the cell surface by aminopeptidases, endopeptidases, exopeptidases, and dipeptidases present in the brass border of the mucosal cells. Some di and tripeptides are actively transported into the intestinal cells and hydrolyzed by intracellular peptidases with the amino acids entering the bloodstream. Protein absorption. At least 7 different transport systems transport amino acids into the enterocytes. 5 of them are dependent on sodium, as they co-transport amino acids and sodium in the similar fashion to the co-transport of sodium and glucose. 2 of them require chloride ion in addition to the sodium ion. The rest 2 transport systems are independent of sodium ion. Dipeptides and tripeptides are transported into the enterocytes by a different mechanism which utilizes H+ ion instead of sodium ion. Inside the intestinal mucosal cells, dipeptides and tripeptides are hydrolyzed by intracellular peptidases into amino acids. Amino acids released inside the intestinal mucosal cells as well as amino acids absorbed from the intestinal lumen and from the surface of the intestinal mucosal cells are transported from the enterocytes to the interstitium by at least 5 different transport mechanisms. Two of them are dependent on sodium and the rest three are not dependent on sodium. Absorption of amino acids is rapid in the duodenum and jejunum, but slow in the ileum. Approximately 50% of digested protein comes from the ingested food, 25% from proteins in digestive juices, and 25% from disformated mucosal cells. Only 2 to 5% of protein in the small intestine escapes digestion and absorption. Some of this is eventually digested by bacterial action in colon. Congenital defect in the mechanism that transports neutral amino acids in the intestine and renal tubules causes heart nerve disease. A congenital defect in the transport of basic amino acids causes cystinuria. Fat digestion. A lingual lipase is secreted by Ebner's gland located on the dorsal surface of tongue. Stomach also secretes the lipase, which is of little importance except in pancreatic insufficiency. The lingual lipase digests as much as 30% of dietary triglycerides. Most fat digestion begins in the duodenum, and the key enzyme is pancreatic lipase. The enzyme pancreatic lipase acts on 1 and 3 bonds of triglycerides with relative ease and on 2 bonds at very slow rate. The principal products of its action are free fatty acids and 2 monoglycerides. The enzyme pancreatic lipase acts on lipids which have been emulsified. Its activity is facilitated when an amphipathic helix, which covers the active site like a lead, is bent back. Polipase, the protein present in the pancreatic juice when binds at carboxyl end of pancreatic lipase, facilitates opening of its lid. Another important enzyme present in the pancreatic juice is bile salt activated lipase. Unlike pancreatic lipase, bile salt activated lipase hydrolyzes cholesterol esters, esters of fat soluble vitamins, phospholipids, and triglycerides. Most of the dietary cholesterol is present in form of cholesterol esters. Cholesterol esters are hydrolyzed by cholesterol esters hydrolases. Fats are relatively insoluble and incapable to move across the unstirred layer to reach the surface of the intestinal mucosal cells. They are emulsified by detergent-like actions of bile salts, lecithin, and monoglycerides after contraction of gallbladder, leading to formation of micelles, which are cylindrical aggregates containing free fatty acids, monoglycerides, and cholesterol in the center. Micelles are readily soluble and move down the concentration gradient across the unstirred layer. The lipids diffuse out of these micelles and a constant aqueous solution of lipids is maintained in contact with the brass borders of intestinal mucosal cells. Fat absorption. Previously, it was believed that lipids enter enterocytes by passive diffusion, but recent evidence suggests that they require carrier proteins. The fate of fatty acids inside the enterocytes depend on their size. Fatty acids containing less than 10 to 12 carbon atoms are readily soluble and they pass through the enterocytes unmodified and are actively transported into portal blood. Fatty acids containing more than 10 to 12 carbon atoms are insoluble. They are esterified to form triglycerides. Some absorbed cholesterol is esterified to form cholesterol ester. Triglycerides and cholesterol esters are coated with protein, cholesterol, and phospholipids to form chylomicrons. Chylomicrons are soluble and are extruded out of the enterocytes by exocytosis and they enter lymph. Inside the intestinal mucosal cells, most of the triglyceride is formed by acylation of two monoglycerides and it takes place in smooth endoplasmic reticulum. Some triglyceride is also formed by acylation of glycerophosphate. Glycerophosphate is the product of glucose metabolism. Some of the glycerophosphate is converted into glycerophospholipid which takes part in formation of chylomicron. Chylomicrons are extruded out of the enterocytes by exocytosis at the basal and lateral surface of intestinal mucosal cells. Acylation of glycerophosphate to form lipoproteins takes place in rough endoplasmic reticulum. Carbohydrate moieties are added to the proteins in Golgi apparatus and finished chylomicrons are extruded out of the enterocytes at basal and lateral surface of intestinal mucosal cells. Absorption of long-chain fatty acids is greatest in the upper parts of small intestine. On a moderate fat intake, almost 95% or more of the ingested fat is absorbed. Short-chain fatty acids containing 2 to 5 carbon atoms are produced by bacterial action in colon on complex carbohydrates, resistant starches, and other components of dietary fiber. Absorbed short-chain fatty acids are metabolized and make a significant contribution to the total caloric intake. Pancreatectomized animals and patients with diseases that destroy the exocrine pancreas have fatty, bulky, clay-colored stools, and the condition is known as steatorrhea. In today's lecture, I have discussed digestion and absorption of carbohydrate, protein, and fat. For reference, you may follow Ganong's review of medical physiology. Thank you.